Unit 4, Genetics. Objectives. Who is Gregor Mendel and what did he discover? How can Punnett squares be used to predict the characteristics of offspring? Why do humans pose particular problems when studying genetics? What are some important genetic conditions in humans? Genetics is the study of heredity. Genetics explains the process of inheritance and why there are variations between offspring from one generation to the next. Virtually every culture in history has attempted to explain observed inheritance patterns. Understanding inheritance patterns is especially important in agriculture, animal husbandry, and medicine. There have been such great advances in the study of the patterns of inheritance in the last decades that it is certainly one of the most dynamic and interesting topics we can cover. Who doesn't have some curiosity about their biological blueprint? To begin our discussion, let's identify two factors that determine the characteristics of an individual. Heredity is the traits that an organism inherits. Environment are all the outside forces that act on an organism. So the question is, which is the biggest influence? It's the nature versus nurture debate. Our understanding of genetics is influenced by Gregor Mendel. Gregor Mendel was an Austrian monk who investigated inheritance at the organism level in the 1860s. Part of his conclusions were that plants transmit distinct factors to offspring. When we look at his work, factors we now call genes, which are found on chromosomes. Mendel worked with the pea plant for several reasons. The boss monk of the monastery would not allow him to work with animals because he believed it would be an insult to God. Peas have a wide variety of characteristics that are easily seen: short versus tall, bushy versus climbing, yellow seeds versus green seeds, smooth seeds versus wrinkle seeds, for example. Peas can be cross-pollinated very easily. Many generations can be observed in a short time. We call that a short generation time. And a small area in which to grow peas works well, like a small garden plot. So Gregor Mendel had a useful model organism. His was the pea plant. Later we'll see others. In Mendel's experiments, if he took a plant and allowed it to self-pollinate, it always produced offspring with traits that were the same as the parent. For example, seeds from tall plants always produce tall offspring, or plants with yellow seeds produce yellow-seeded offspring. He also experimented by crossing plants with contrasting traits. He could cross tall plants with short plants, or yellow-seeded plants with green-seeded plants. In his experiment crossing tall plants with short plants, he found that it always produced tall plants, whichever one supplied the pollen. Looking at the anatomy of the pea plant, specifically the pea flower, we can see how he was able to control crosses by either cutting away anthers and brushing pollen on from another plant, or allowing this plant to self-fertilize. Mendel then could cross plants that were produced from experiment 2. Experiment 2 were those that had contrasting traits. He called the original cross in experiment 2 the P1, or first parental generation, and he called the generation produced from P1 cross F1, or the first filial generation, called the generation produced from the cross of P1 plants F2, or the second filial generation. Now, the second filial generation provided some amazing and unexpected results. So, if we're looking at this parental generation here, green parents with yellow parents of their seeds, so when they were crossed, all of the offspring had the yellow seeds. That's the first filial generation. Now, the second filial generation, he found that he got about 75% of the offspring had yellow seeds, but 25% had green seeds. So this trait had skipped a generation. He saw this with several other traits as well. So Whenever he blended tall and short plants, the first filial generation, all were tall. If he allowed those to self-fertilize, then again he saw this 3-to-1 of 3 tall to 1 short. Now, part of the idea at the day was this idea of blending inheritance. So the blending model would predict that if you cross a tall plant with a short plant, you get a medium plant. And if that was to self-fertilize, you would get all medium. That's not what he saw. So, besides looking at tall versus short, he also looked at pod-shaped, whether it was inflated or constricted, the seed shape, round versus wrinkled, seed color, yellow versus green, the flower position, whether it was axial or terminal, flower color, purple or white, and pod color. In all cases, he saw that the second filial generation produced this same approximately 3-to-1 ratio of what he called a dominant trait versus a recessive trait. What we want to note here too is that he did this with thousands of plants so that he had statistically valid data. So keep in mind that Mendel didn't have any knowledge of chromosomes or cell division or other things that we do today. But based on his observations, he determined that the following were true: that each pea plant has 2 factors for each trait, we now call these genes, and that these factors segregate or separate during the formation of gametes. Now, we just talked about cell division. Where did we see this concept? Mendel also determined that each gamete contains only 1 factor from each pair of factors. This should also give you a clue as to which type of cell division this is. And furthermore, Mendel said that fertilization gives each new individual 2 factors for each trait. All of this is part of the law of segregation. Another part of Mendel's hypotheses was his principle of dominance and recessiveness. He used letters to indicate each allele, and a capital letter symbolizes a dominant allele, a lowercase letter symbolizes a recessive allele. An allele is a variation for each trait. We'll get into that more as we go along as well. If you have a dominant allele or dominant trait, it's going to mask the expression of the alternate, which is the recessive allele or trait, when both are present in an organism. So, if you have an organism, such as pea plants, that has 2 factors or genes for each trait, if you have 2 dominant alleles, then that plant will be dominant. If you have 2 recessive alleles, it will show the recessive trait. But if you have 1 of each, the dominant allele masks the recessive one. So, 1 factor or gene in a pair may mask the other and prevent it from having an effect on outward appearance. For example, in peas, tallness is dominant and short is recessive. So if we had an uppercase T to represent tallness, lowercase representing the recessive or short allele, if an organism has uppercase T, lowercase t, that's going to look tall, even though it has both traits. This is a hybrid. So Mendel had no knowledge of meiosis, which is the type of cell division that helps explain his findings. But he did realize his results were attainable only if the sperm and egg contains every possible combination of factors. This gave rise to the law of independent assortment. That is, that each pair of factors assorts independently without regard to how the others separate, and that all possible combinations of factors can occur in the gametes. Gametes, as we said earlier, were sex cells, so in humans this was eggs and sperm. Even though Mendel didn't know about chromosomes, having a workable definition of some of these terms will help us in our understanding of genetics. In peas and in humans, chromosomes come in pairs called homologous chromosomes. One member of the pair is inherited from the mother, and the other is inherited from the father. We looked at this concept earlier in cell division. Homologous pairs have certain characteristics. Both members have the same length and centromere location. Both carry similar types of genes, and alternate forms of a gene for a trait are called alleles. We just looked at this concept of dominant or recessive alleles. And alleles are always on the same spot or gene locus. When we're looking at organisms such as ourselves and pea plants, we are diploid organisms. And that is that we have a pair of homologous chromosomes that contain two alleles for each trait. One allele is on each member of the homologous pair. Phenotype is an individual's actual appearance. This may include physical characteristics, microscopic or metabolic characteristics. So this is the phenotype, physical appearance. For pea plants, this is things like if it's tall, if the flowers are purple, etc. For humans, this would be whether or not you have freckles, whether or not you have a bent little finger. The genotype describes the alleles carried by the chromosomes that are responsible for a given trait. Looking at homologous chromosomes, keeping in mind that one member is inherited from one parent, the other is inherited from the other parent. So this is a homologous pair of chromosomes. So notice how they have the same length. This small indentation here is the centromere. During cell division, that's where those spindle fibers attach. These letters here are representing alleles, and this area that they're shown is the gene locus. And see that they have the same types of information in the same order. So the upper and lowercase letters represent either a dominant, which is an uppercase letter, or a recessive form of that gene, which is lowercase.